In the autumn of 2001, a series of lethal envelopes unleashed a new form of terror, compelling investigators to forge an entirely new discipline: microbial forensics. This unprecedented challenge demanded a scientific revolution in crime solving, forever altering the landscape of biosecurity. Letters postmarked in Trenton, New Jersey, began reaching prominent media outlets and the offices of U.S. Senators Tom Daschle and Patrick Leahy. This deliberate dissemination of a deadly pathogen marked a chilling escalation in domestic threats. The attacks tragically claimed 5 lives and sickened 17 others, causing widespread panic. The U.S. mail system faced partial shutdown, underscoring the profound societal disruption a biological agent could inflict. The powder within these envelopes was identified as highly purified Bacillus anthracis spores. This hazardous evidence necessitated unprecedented protocols for collection, handling, and strict chain of custody to ensure both safety and evidentiary integrity. Short, threatening messages accompanied the spores, amplifying public fear and the psychological impact of the attacks. Investigators meticulously preserved every physical clue: paper, ink, fibers, and adhesive tape, recognizing their potential forensic value. Specialist teams, often working in hazardous conditions, secured samples from contaminated sites. Every transfer was rigorously documented, and evidence was split for parallel testing across multiple accredited laboratories, ensuring robust validation. Microbial forensics emerged as a critical discipline, shifting the investigative paradigm. Instead of human fingerprints, scientists sought the unique biological signatures embedded within the Bacillus anthracis spores themselves. Analysts observed rare colony variants, termed morphotypes, within the cultured anthrax. These subtle phenotypic differences acted as crucial genetic breadcrumbs, guiding the investigation toward the pathogen's origin. Targeted genetic differences were meticulously identified, distinguishing the common laboratory strains from the highly distinctive material found in the letters. This molecular fingerprinting was pivotal for source attribution. These unique genetic markers provided a critical breakthrough, pointing investigators toward a specific seed stock of Bacillus anthracis held within a U.S. biodefense laboratory. This narrowed the potential source considerably. Agents then meticulously audited the distribution of aliquots from that specific seed stock. This exhaustive process created a precise shortlist of individuals and facilities with access to the identified material. Forensic analysis of spore contamination in Postal sorting equipment allowed investigators to map the mail's precise path. This Postal traceback identified key facilities and potential points of origin for the letters. Beyond the biological, traditional physical forensics played a vital supporting role. Analysis of envelope types, printing methods, stamps, and handling patterns provided crucial, non-biological clues, corroborating other findings. Initial public suspicion, fueled by media speculation, unfortunately fell upon Dr. Stephen Hatfill, a former biodefense researcher. He was later definitively cleared of any involvement and received substantial compensation. As the investigation progressed, attention shifted to Dr. Bruce Ivins, a senior microbiologist at the U.S. Army Medical Research Institute of Infectious Diseases, USAMRIID. He had direct access to the identified anthrax stock. Investigators meticulously reviewed lab logs, access card data, email communications, and Dr. Ivans's movements around the times of the mailings. This comprehensive contextual data was crucial for building a circumstantial case. Crucially, the rare morphotypes identified in the letter anthrax precisely matched subcultures directly tied to Dr. Ivans's laboratory flask. This genetic consistency strongly correlated with his documented access to the source material. Search warrants were executed, gathering digital files, personal notebooks, and laboratory controls. The convergence of genetic evidence and contextual information significantly tightened the investigative focus on Ivins. In August 2008, a pivotal turning point occurred when Dr. Ivins died by suicide, just as federal prosecutors were preparing to file criminal charges. Consequently, the criminal case never proceeded to trial. In 2010, the U.S. Department of Justice formally concluded its Amerithrax investigation, publicly naming Dr. Bruce Ivins as the sole perpetrator of the anthrax attacks. This marked the official end of the criminal inquiry. A subsequent independent scientific review by the National Academy of Sciences affirmed that the genetic evidence strongly supported the conclusion regarding the anthrax source. However, it cautioned that genetics alone could not definitively prove guilt without corroborating contextual evidence. The anthrax letters case profoundly matured the field of microbial forensics. It spurred the development of better reference databases, advanced genotyping techniques, and more rigorous evidence handling standards for biological materials. In response to the attacks, male security protocols were drastically enhanced. High-risk male streams began undergoing irradiation and targeted screening, fundamentally altering how sensitive mail is processed. The incident also strengthened public health preparedness. Hospitals, diagnostic laboratories, and law enforcement agencies established more robust, rapid reporting systems and conducted joint drills to improve coordinated responses to bioterrorism. Laboratory oversight was significantly tightened, particularly for facilities handling select agents. New regulations, stringent inventory controls, and regular audits were implemented to prevent similar incidents. The integrity of biological evidence became paramount. Chain of custody practices were expanded to meticulously track not only physical materials, but also associated metadata, ensuring transparency and accountability. The attacks unfortunately led to thousands of white powder scares nationwide. This necessitated the rapid evolution of triage protocols and public risk messaging to manage widespread anxiety and prevent overreactions. The resolution of the Amerithrax case underscored that scientific method, not magic, drives complex investigations. While genetics narrowed the field, classic investigative techniques and logistical analysis provided the crucial contextual framework. Ultimately, the anthrax letters case stands as a stark reminder of both the immense power and inherent limits of forensic science. Convincing conclusions require the convergence of robust evidence from multiple, independent investigative lanes. For a deeper dive into the intricate intersections of science, law, and criminal investigation, subscribe to the AI Criminologist. Join our community of researchers, professionals, and curious minds as we unravel complex cases and explore the evolving frontiers of forensic analysis. Your engagement fuels our mission to educate and inform.